the Dementia Researcher podcast, talking careers, research, conference highlights, and so much more.
Today we're going to be recording on location from Lisbon in Portugal and going to share highlights from the first 3 days of this year's ADPD conference. This event brings leading experts together to unravel the mechanisms of diseases and to talk about the progress made on new treatments. Hello, I'm Dr Amanda Heslegrave and I'm a senior postdoc in the UK Dementia Research Institute at University College London. I lead a programme of work and research to deliver fluid-based biomarkers for Alzheimer's disease, but also all other forms of neurodegeneration. Now, joining me today, I have 3 more researchers and they're going to share their favourite talks and posters from the first 3 days of this conference, which is probably one of the biggest events in the calendar for neurodegenerative disease after AAIC. So I'd like to introduce Dr Ariana Gatt and Dr Ian Harrison from University College London and Jess Tobin from Alzheimer's Research UK. So hi everyone.
Hello.
We're going to start with a round of introductions and to give you guys a chance to talk about any research that you will actually be presenting this week. So Ian, why don't you go first?
Cool, so thanks so much. So my name is Ian Harrison. I'm a senior research fellow at University College London and I work in the Centre of Advanced Biomedical Imaging at UCL. So my lab works on the lymphatic system, which is this fluid clearance system in the brain. Particularly we're interested in the role this system plays in propagation of proteins, so alpha-synuclein and tau mainly, and how it facilitates spread and clearance of those from the brain. So this week, yesterday actually, I presented a talk in one of the alpha-synuclein sessions here, talking about some of that work, sharing some of the data that we acquired recently on an CORIN4 inhibitor, showing that it worsens the effects of alpha-synuclein propagation in our mouse models.
Fabulous, thank you very much. Ariana?
I'm Ariana Gatt. I'm also a senior research fellow at UCL. I work at the Tamara Nashley lab, and we are mainly a neuropathology lab, so we do a lot of histochemical staining, predominantly on postmortem brain tissue. And my interest lies in RNA binding proteins belonging to this family called HNRP, and their involvement in disease, more specifically fronto-temporal dementia. So there's a famous RNA binding protein called TDP43, which is very well known in its role in FTD, but we're more interested in it what I like to call protein cousins, so the ones belonging to the same family, but at the moment not as well studied as FTLD. And I've got a poster here looking at how this HNRP family is altered even by single-cell RNA-seq data in different subtypes of frontotemporal dementia.
Okay, thank you very much. Now, Jess, can you tell us about yourself?
Hello, yes, so I'm Jess Tobin, so I'm the Science Communications Manager for Alzheimer's Research UK. Obviously, I'm not presenting anything this year or have any posters, but there's about 5 of us from Alzheimer's Research UK that have come over for the conference. As you mentioned, Amanda, it's a really key conference in terms of neurodegenerative diseases, and there are a number of quite large talks that we're expecting over this week. So some phase 3 trials, which could potentially get some kind of wider coverage, but then also a lot of our researchers are presenting and have posters. So it's really great to come along to events like this. There's so much going on in the field, and there's been a massive kind of upheaval in the number of areas that are being investigated at the moment. So each year we kind of come to conferences, it seems the program is expanding. So it's really fantastic to be here.
Okay, thanks very much. So with the introductions done, let's get to the highlights. So for anyone new to the podcast, let me explain the format. Now this is quite simple. Essentially we go around the guests 3 or 4 times, allowing them to talk about what they've seen, what has stood out to them, what they found most interesting, and then just give a summary and what they've taken away from that. So I am going to go to Jess first this time.
Yeah, Brilliant. So, as I kind of mentioned, this year is a really big year for Alzheimer's disease, in particular in terms of the first disease-modifying treatments starting to come through. Obviously, we've already had them approved in some countries like the US, but we're now waiting for the MHRA decision for lecanemab. And obviously, for people affected by dementia, this is a massive turning point. This is the first ever time we've been able to prove that drugs can modify or slow down the progression of Alzheimer's disease. And so there was a symposium hosted by the Alzheimer's Disease Drug Discovery Foundation yesterday, in which our executive director of research and partnerships, Susan Collas, sat on the panel. And that was a really fantastic discussion looking at the kind of the progress that we've made over the last 40 years, even within the last 10 years. I mean, when I started Alzheimer's Research UK just 4 years ago, the idea of having a disease-modifying drug seemed quite a way away, and it didn't seem something that was kind of tangible. And within the last year especially, it seems like there's just been this massive momentum and drive forward. So they hosted a fantastic panel discussion looking at the wider kind of therapeutic options. And as we know now, these kind of removal drugs targeting mostly amyloid, but also going into tau, are now just a small kind of proportion of the drugs that make up the pipeline. And Geoffrey Cummings later presented just after, and they were talking about how in his Cummings report that just 25% of the drugs now in the pipeline target those amyloid and tau plaques and tangles, and so about 75% of those drugs are looking at other mechanistic areas. And so that's such a promising statistic for people affected by dementia, and also in the wider field, especially when we're starting to look at kind of combination therapies and outcomes patients. So it was a really fantastic overview of the wider field at the moment.
Thinking about, you're talking about the disease-modifying treatments and how exciting that is. I think one of the highlights for me here is listening to people talking about how we then diagnose better, and the possibilities of blood-based biomarkers, for example, which of course I'm really interested in, but also the care and attention that's got to be made so that we know what the biomarkers are telling us and that we use them in the right context. And I think that there's still quite a lot of there's still quite a lot of work to do on that, let's be quite honest.
Yeah, exactly. And in terms of the kind of fundamental research we've come so far in terms of identifying biomarkers and seeing which ones hold the most promise and which work, especially in relation to kind of gold standard like PET. But you're right, it's that integration into real life communities and settings, and that's why things like the blood-based biomarker challenge that ARUK are launching with Alzheimer's Society, and then NIHR, which will be looking at embedding kind of blood-based and fluid biomarkers into NHS and real-world settings is within hopefully 5 years is really important.
And then Ian, tell us.
I'm gonna go for one of the talks I saw yesterday. Moving completely away from biomarker stuff, but I felt a bit bad for the guy because it was the last session of the day, and you can tell that everyone was exhausted, and it was in a very stuffy room, but I thought the science was really exciting. So it was from Lin-Hui Tsai's group at MIT, and it was presented by Mitchell Murdoch, who was a PhD graduate student in the lab. And what they did was that it was an animal study, and what they did was that they exposed the animals to this 40 hertz sensory and visual stimulation. So basically they put the animal in a box, flashed a light in its eyes, and gave this 40 hertz frequency sound, and it caused this massive upregulation of the glymphatic system. When they did this in mice models of the 5 times FAD mouse, which is an amyloid model, they saw a reduction in the amount of amyloid that was accumulating in their brains just from exposing the animal to a flashing light and a sound. The paper was actually published last week in Nature, so it's the first time that the work has kind of been presented at a conference. They haven't presented it until now. But yeah, it was a fascinating talk, and they kind of looking a bit more about the mechanism of that. They'd identified a neuropeptide which is involved in that they show that there's an upregulation of after this multisensory stimulation. But it was a fantastic talk. It's showing quite an odd therapeutic intervention and how that can have a massive effect in terms of just brain function and CSF dynamics.
Has there been lots of lymphatic presentations at this?
Not too many. I think there's still a couple of posters that I need to go and check out. But it's one of those fields where we're often spread about the different categories. So I was in the synuclein talk. That talk was in a kind of an amyloid therapeutic session. So they're kind of dotted around, but if you look for them, you can find them.
You need to find exactly where your hook is and like get in there. Okay. Then shall we move to you, Emily?
Yeah, I'm sort of going to link on to what Ian just said. I was at an A-beta session yesterday as well, and there was a talk by Andre Obenaus from Uni of Irvine, California, and they did this really interesting study, I thought. I'm quite into this whole traumatic brain injury field and how it links to neurodegeneration, and so they gave this talk where they had these juvenile and midlife mouse models of TBI. And what they found is that even an early on juvenile traumatic brain injury incident can cause Alzheimer's-like changes much later on, which is really interesting, like behaviorally, cognitively. They also see similar changes in the cerebrovascular staging and vascular uncoupling. And what's interesting is massive disruption of the blood-brain barrier, similar to what you see in Alzheimer's disease, but in these juvenile mouse models of traumatic brain injury, which I thought was quite interesting.
So, okay, all right. Well, I'm going to talk about, just briefly, the plenary, one of the plenary lectures that I really enjoyed on Tuesday, which was the genetics of neurodegenerative diseases, and that was from rare populations to common variants, and that was Rita Guerrero. And actually, I do know her because she used to work at ION, and I think the thing about her work that strikes me is that by looking at something that doesn't seem even related to what we're doing, like macular disease and TREM2, then you find, okay, there's dementia in this disease. Could it be? Oh yes, it is. It is actually a risk factor for a very common disease, which is sporadic AD. And I think that the amount of information that we found from studies like the ones that she does with the different kindreds, etc., is invaluable to, and it will be invaluable moving forward for the rarer diseases still that, or when we try to put together a complete kind of like risk score to add to a screening program, for example. Like, I mean, each one of your variants adds up to something, and that leads you then to perhaps take that, not statin, but brain statin. That's what I'm going to call it, a brain statin, so that we can like protect ourselves before anything even goes wrong. So then we move back to Jess, some further hope.
Yeah, so kind of to add on to your summary, Amanda. Also, one of the plenary lectures on Tuesday. Hopefully, I don't butcher the name. By Henne Helsted, from the Netherlands. So she is doing the 100+ study, which is looking at the centenarians who remain cognitively healthy. And I thought it was such a fascinating take on a way to kind of do one of these longitudinal studies, because I think a lot of them which work with humans and are like clinical trials are usually looking at kind of like disease pathology and what goes wrong. And it was quite nice and refreshing to have a study looking at what goes right, and some of the findings that are already coming through confirm some of the kind of beliefs that we hold around kind of risk reduction messaging. So a lot of them kind of didn't smoke as much, or didn't like had similar levels of like increased levels of exercise and social attainment and all these other things. But then there were also some really interesting factors that they pulled out, like they had a greater sense of optimism, which I thought was really sweet, and
Here.
yeah, exactly. And they pulled out some of the, I don't know if any of you went, but they pulled out some of the quotes for some of these centenarians at the end, and they had some really hilarious kind of life advice, like don't date too many men, and all of these other factors, which I thought was a really kind of I don't know, like upbeat and positive way of looking at research, when a lot of these kind of studies that we're looking at, unfortunately, a lot of the time we're working with people that are really severely affected, and It's obviously a really devastating condition for a lot of people, so I thought it was a really nice kind of lightening topic. And I think there's the theme of aging is running through this conference quite a lot, and Miranda Orr yesterday in one of the symposiums spoke about these senescent cells in the body which are damaging, and the possibility of kind of, if we can clear these away potentially in people's 30s and like reduce the significant effects of aging, that we can kind of try and prevent a lot of these things that go wrong downstream. So I just thought, I thought she gave a fantastic presentation, and it was really, really cheerful, I thought.
One thing that struck me about the Hene Holster talk was the socioeconomic status, that was an important factor, that they all did not come from deprived families. So that was one thing.
Yeah, we're seeing that a lot, especially with Lancet, so the 12 modified risk factors. A lot of these things like education, educational attainment, the things that affect early on, they have links into socioeconomic factors. And so that's something our policy team are working quite closely with the government on.
If you boil it all down to something, it was that they had less genetic risk factors and more protective genetic factors. So there's a certain amount of luck there, I think.
It's entered this really fantastic slide looking at how your genetics influence your overall health. And I've written it down so I didn't forget. They said something like roughly 20% of your genes you're reliant on when you're about 60, and that increases to about 70% by the time you reach 100. So those lifestyle effects do have quite severe implications, but as you get older, they start to drop off, and you do rely more on your genes. So there is that real necessity for gene targeting as well.
And they looked brilliant as well, didn't they, in their photos? I wouldn't have even said 80s like they were saying. I was going more 70s, and I could only hope.
So.
Right. And then I think we moved back to Ian to see what more you've got for us.
Another talk I wanted to highlight was selfishly it was in my session, but I thought it was the speaker before me, Maria Spilantini, who everyone probably knows from kind of history with alpha-synuclein in tau and everything. But her lab has been developing this new animal model. So she's particularly interested in synuclein propagation and how the potential for it to move from the gut and it starts to accumulate in the kind of epithelial cells of the gut, move up the enteric nerve and then into the brain. So they've developed this new animal model where where the animals express the 1 to 120 form of alpha-synuclein, but it's driven by a promoter which is only expressed in the gut endothelium. So these mice only get alpha-synuclein in the cells of the gut, and then their group's starting to study its travel up into the brain, via the vagus nerve, and doing some vagotomies and things to see how that route is affected, if it's lateral or contralateral. Really interesting stuff, showing like the primary route that alpha-synuclein starts to seed in the brain, and then they see once it's there, then it spreads to other areas that are synaptically connected. But it's really lovely. We're kind of used to seeing the similar animal models, especially in my field, in the propagation field, where you kind of induce with a prion-like promoter or a prion-like protein. But it's nice to see that there's still movement in the kind of animal model development, this stuff. And it's really nice at the end she was saying, we want to give these mice to people. If you want to collaborate, if you want to use them, let us know, we will send them.
I actually think it's incredibly brave to set out on a project to make a new model, doing something that's quite different, and then wanting to share it is, I realise that that's written into a lot of grants now, that you have to share the materials that you make, and that's right, that's only right, that money should go to do that. But like making something completely new that's going to be useful to the field, I think is a real bonus.
Yes. So I'm going to move back to the resilience talk for a tiny bit. I didn't actually go to the plenary yesterday because I was at another session, but this morning I did go to the resilience session. And it was a really great, uplifting way to start the day, positive news, as Jed said. And I heard there was a talk about this 100+ project. And I did notice something. I don't know if it was sort of statistically important, but a huge part of the cohort they have is female. I don't know if did you guys notice that? 72% is female, which is interesting. I didn't know if this was discussed yesterday in the plenary, but it's interesting because also we know that maybe there's a female correlation to more Alzheimer's disease probability. So maybe it is something about a switch.
Yes, but women have a longer life expectancy.
Exactly.
In general. So it's a weird relationship.
It's true, right? But I thought that was interesting. But there was loads more talks in that session that were amazing. And I think a lot of that talk that was sort of a common thread throughout was the inflammation aspect, that potentially what happens in these super healthy centenarians or the resilient population is they might have as much amyloid load, but then the inflammatory response kind of different to a level that is protective. And there was the last talk of that session was by Beth Stevens from the Broad Institute, and she discussed this highly deeply phenotyped Finnish cohort, which is a brain biopsy live cohort. So it's a sort of, it's amazing, right? So an early Alzheimer's disease staging cohort, and they're doing lots of omics on it. So obviously single cell RNA-seq data, bulk-seq data. And again, it's mainly a sort of microglial study that they're looking at. But one thing that I did pull out from it that was mentioned yesterday, actually by another talk by Andrew Octavian Sasmita from the Max Planck, is that there seems to be a thing now that oligodendrocytes produce beta amyloids. So in a lot of single cell data, they seem to be picking up that oligodendrocytes have a high amyloidogenic gene expression, which is quite interesting.
And unknown to me.
And quite new, but so I think it's kind of cool at ADPD where, yes, there's obviously the biomarkers and what's happening in the clinic, but what's really, from a fundamental biologist point of view, is to pick out these things that are fairly new that in the future will be,
And one of the things you're talking about resilience, it does come up a lot in what we do, is why do the people who have lots of amyloid, some people who have lots of amyloid, why do they not get dementia? Why does that? Why are they spared? But also it's really important in the world of disease-modifying therapies. Why would you give someone a drug they don't need? How do you know they don't need it? It's something that's going to be a massive question.
And that's quite interesting. The talk that was just on the symposium hosted by ESI, which Jeffrey Cummings spoke at, he was talking about what is clinical meaningfulness for people diagnosed with Alzheimer's disease and going into clinical trials especially. And there was a really interesting slide in which they highlighted the difference between FDA approval in America and EMA approval in Europe, in which they said that, aducanumab was approved by the FDA, but it was not approved, I can't think of the word then, not approved by EMA. And that was because the FDA had criteria where they would accept biomarker diagnosis in lieu of clinically significant cognitive impairment. So for people that showed positive amyloid clearance on PET, that means that they approved that drug as clinically meaningful, whereas the EMA would not accept biomarker testing in lieu of any kind of, for pre-symptomatic patients. And so that's a really interesting point that different kind of government bodies and agencies will have different kind of criteria around what they accept as clinically meaningful.
It was a fascinating talk as well from a kind of like basic fundamental scientist point of view. Seeing, Jeffrey Cummings went through the numerous different cognitive scores and all of the different scales that you read about in papers, but he kind of talked through them and said, this is what this one highlights. This one's, particularly sensitive to MCI, for example. And you don't, from my point of view, as an animal scientist, I don't know these things, but it's kind of, it puts the papers that I read and when you read about the clinical trials data, it kind of puts that into a bit more perspective.
Yeah, and I think that maybe there needs to be more education for all of us on what those papers mean, because it is quite hidden sometimes what the meanings, and we can't really interpret them properly. But I think, for biomarkers, it's absolutely crucial. But also what's crucial, I think, is the people who run the trials making the samples afterwards available to everybody else. Not just their results, but those are precious samples of people who've been treated with drugs that could tell us a lot of more information. It's probably not in the remit of what I'm doing here, but something that I feel quite strongly about.
Yeah. I've also seen a couple of posters that I think are interesting with regards to, and I think this is a field that we should be more open to discussing and looking at, is the effect of medication on the transcriptome in the body, and that could be neuroprotective or harmful. So I think that's really interesting. And even this morning there was a talk by Howard Phillip who was discussing aging and dementia, and he's the chief scientific officer of the Alzheimer ADDF. And he was saying about how now 40% of AD medication is actually repurposed drugs, and 40%, which is amazing, right? And he brought up metformin, which is, as we all know, an anti-diabetic drug, but has these amazing neuroprotective effects that nobody's really looked at, maybe delved in as much.
So there's a lot of research at the moment, especially in the Parkinson's space, and AD, looking at the G1P1 agonists. Yeah, so that's kind of similar to, Saxenda and all those other things, Myovi and all the Ozempic, the weight loss drugs. They're thought to have major protective effects.
Is this via their effects on metabolism?
Yeah. So I heard an interesting tidbit the other day. I might have to fact check this, but apparently they found, they started finding these drugs because there's a lizard that exists that only eats once a year. Yes, and they were like, how can it maintain blood sugar? Why does it not die? How does it kind of like modulate insulin and all these other things? And they found it had a really high number of these G1P1 receptors. And so then they started looking at agonists for these receptors, and they developed these group of drugs, which now is diabetes drugs, but they're looking to have loads of other effects. And as we know, glucose metabolism goes quite wrong in a lot of neurodegenerative diseases. So it's a really interesting area of science at the moment.
And they might have effect on vasculature and all of these things, right? It's so multifactorial. I think it's amazing.
Does anyone want to just...
There was a really fantastic talk. So obviously ARIA, so amyloid-related imaging abnormalities, is a big area of discussion at the moment, especially when looking at these amyloid-targeting drugs like lecanemab and donanemab coming through, especially as ARIA, unfortunately, seems to affect people with APOE4 status the most. And obviously they're a population that are more at risk of like late onset or sporadic Alzheimer's disease. And up until very recently, even now, the mechanisms of why ARIA happens aren't fully understood. And Donna Wilcox, so she's based at the Indiana University School of Medicine in the US, did some studies and proposed a kind of novel mechanism. And as we've all kind of discussed, inflammation and immunity is a really big topic this year, and it all seems to relate back to that. But she proposed a really novel mechanism around kind of the complement system, so C1, and so how the anti-amyloids binds to amyloid beta in our vessel walls, which then allows kind of the C1 complex to form and membrane attack complex, and triggers those downstream inflammation processes. But then she also proposed that it's possible that APOE has a role in binding to C1, and so if it's a healthy version of APOE, that could more actively bind to C1 complex, but if those that have APOE4, it doesn't bind as readily, potentially meaning that they have much more inflammation and much more ARIA in the brain. And so that potentially has massive therapeutic implications in terms of being able to develop these drugs for everyone affected or people with that APOE4 status. So it's really exciting that she was part of a number of talks looking at potential ARIA mechanisms. And considering we've only kind of had these drugs around and done these trials in the last couple of years, it's incredible the speed of science nowadays and how quickly these mechanisms are being investigated and proposed. And so I just thought that was a fantastic talk.
The whole ARIA scenario is really important, and the need for some sort of test or indication of who we need to monitor more carefully, who shouldn't get the drug because it will be really bad for them. I think those things are really important. But the thing about the science speeding up, I honestly think that COVID showed us how quickly it can move when it has to. Yeah. And now we've got disease-modifying drugs. I feel like the whole field has been like, it's like, and I mean, and it's great. I don't think there's anything wrong with how we were working before, but I also think that when you can't see that we're able to do anything, it is much harder to have the momentum. And I feel like we've got it now.
And the technologies, right? So now it's amazing for me to look at these posters, everyone doing single cell and everyone's doing more RNA-seq. And that's great because that's data that's available to everyone and we can all, compare and contrast. And it also makes science more global, I feel, because you can compare the effects that are happening in, say, cases from the US compared to other countries, and we can all sort of just
sharing data
harmonize
easily.
Yeah, exactly. I think that'll be a clue to finding eventually disease-modifying therapies as well.
And I think, at that point, probably the other plenary that was on a Monday that was really good, but I'm not going to claim to be any sort of an expert, was, Koichi Tanaka, who I listened to he's Japanese, but he's a Nobel Prize winner and has done amazing things with mass spec technology and developed lots of things I can't explain really. However, I have used mass spec and it's a very useful tool for, discovery science. We wouldn't know half of the proteins that we look for now without mass spec, but also it's really useful and we do use it for kind of targeted identification of proteins as well, where some proteins are just impossible to find with antibodies or very well, and we can use targeted mass spec. And so I don't really know what he got the Nobel Prize for, but I'm sure it was really good. I don't know if anyone else went to the cryo-EM structures of disease, because the fact that structures can be just slightly different for different diseases is a bit mind-blowing. But what did you think?
Yeah, like you said, it was the plenary yesterday from Michel Godet, and he was talking about cryo-EM resolved structures of tau and alpha-synuclein and amyloid in various different forms of neurodegenerative disease. So what was quite nice is that Michel's group at the LMB published this work. The first tau structure maybe about a decade ago, something like that, 5 years maybe. But what I really liked about his talk is that he was showing like the slowly increasing N number, saying like we've done 12 pure Alzheimer's patients now, we've done another 6 Parkinson's patients.
So it's dependent on brain donation, like so.
Exactly, because in one of the original criticisms of their work was that it was an N of 1. There was a brain sample and the cryo-EM structure of tau was resolved, which was fascinating, but there's still that question, is one. But it was really interesting seeing how he was kind of stratifying all of the protein classes and the different folds, and how slight changes means that you have a completely different way that protein builds up in Pick's disease, for example, rather than all of the kind of rarer neurodegenerative diseases, but they share this kind of common mechanism with protein accumulation. But yeah, fascinating. Yeah, really good.
So has anyone else got any other particular highlights, even just about the conference, not about a talk or anything?
Yeah, I just found, again, the resilience talks this morning really, that session was amazing, I thought. There was another talk by Bradley Hyman from the Massachusetts General Hospital, and they showed this amazing finding, I thought, that they had these tangle-bearing neurons compared to non-tangle-bearing neurons in vitro, and that the non-tangle-bearing neurons have a higher risk of dying than the tangle-bearing ones.
So there's an
Wow.
advantage.
So he wouldn't
Like a
sort of
protective?
say, I'm saying that, a tangle is a good thing, but that potentially sort of the more fibrillar form is potentially
type of
slightly,
protein.
so the non-tangle bearing ones are more at risk of dying, 3 times more or something like that?
Yeah, like that, maybe 30%.
Yeah.
It's a 3.
There was a 3 somewhere. But he also showed something which I thought was very interesting, is that 1 to 2 weeks before dying, these neurons show a slight shift in position. They swell so ever slightly that their neighboring neurons move a tiny, tiny bit. And that is insinuating that that neuron is about to die. I just thought the whole microscopy, imaging way, it was just amazing. Yeah. Really fascinating stuff.
There was one other talk by Michaela Lunn from the University of Ontario, Canada. Ottawa, yes, I can't read my own writing. University of Ottawa, Canada. So she was looking, this was on yesterday, she was looking at nasally acquired viral infections in mice studies and whether they kind of impacted risk of neurodegenerative diseases. So I believe most of her work is based in Parkinson's, but then there was some application to tau and phospho-tau, which is why she was speaking at the tau talks yesterday. But I think this is quite an interesting area for, in general, the public, because there's a lot of talk at the moment around like COVID and things like brain fog, and we don't quite understand yet what that relationship was. And obviously, massive caveat, this is in mice, and it was looking at, so it looked at VSV and H1N1. So H1N1 was the more kind of the peripheral lung type virus. And what was really interesting is they introduced these viruses to the mice, and then they waited, those that survived, they waited 6 weeks to see whether there was any kind of tau, phospho-tau in the brain. And they found for both VSV and H1N1 that there were significant levels of phosphorylated tau compared to placebo, compared to no virus in the mice after 6 weeks, showing that there could potentially be some quite significant long-term neurological impacts from acquiring a virus. And so obviously this is very early research and it's in mice, but this kind of research is really important in us starting to understand the long-term implications of viruses like COVID. So I thought it was a really fascinating talk.
But also, I mean, we acquire infections throughout our whole life. How much of that then appears in our brain as amyloid or something later? How much we don't. There's not a lot we can do about colds and stuff like that, but there will be a certain amount of damage left behind.
We do now know from RNA-seq single cell data that you do get a lot of viral sort of gene-associated pathways upregulated in a lot of neurodegenerative diseases, which again goes to show there's a link there. So that's interesting. Okay. Something that I assume again will be looked at more and more in the future.
Question to all, and we'll go round. From what you've learned over the last few days, what excited you the most, and what do you think is the next big discovery on the horizon? Ariana, first, please. I'm going to put you on the spot.
Thanks for that. I think as a fundamental biologist, I do think that the larger cohorts of data that we're getting is quite exciting. All the omics that is happening, all the RNA-seq data that we're getting, that again will allow for greater collaboration and for finding these little, maybe thus far hidden, factors that are involved in neurodegeneration that will sort of create more and more momentum into looking at maybe avenues that haven't really been explored yet. Like maybe this viral infection, or the fact that maybe it's not just about neurons making beta amyloid, but there's other cells involved in the brain. I think these are the things that excite me the most about conferences like ADPD.
Okay, thanks very much. Ian?
I think one of the talks that most excited me was that glyphosatics talk that I mentioned, and I think it's one of those fields which it's been in the periphery of neuroscience for a while, but is now starting to just play more of a role in conferences like this and in Alzheimer's disease, that kind of disease researchers are starting to come on board and look at this pathway in the brain. In terms of what I think the next big discovery is, I don't know. I think something along the lines of, I mean, there's all of the kind of phase 3 data and all of the those trials data which are now kind of delivering. I think from my perspective, I would really like to see some movement in the kind of lifestyle changes as being testable and tangible things that can be put through trial to see if they have kind of any, tangible effects in terms of reducing disease burden or progression.
Okay, something achievable. Brilliant. Jess?
Obviously, amyloid has come on, anti-amyloid drugs have come on massively, and we're now waiting on, like Ian said, phase 3 trial data from TauRx. But I think from what I've seen so far, what Ian mentioned earlier about that gamma radiation, the 40 hertz clearance, but then also some of the, so there's some small molecule drugs in development for TREM2, and it's these kind of less invasive potential treatments that are potentially coming through, which will allow us to deliver combination therapies. So the lecanemab and donanemab, they're potentially great for a very small subset of people, and they only have a modest effect. And if we're to develop disease-modifying treatments for much more of the population, then we need those combination therapies. And so, I think there's a lot of promise and a lot of different areas in the program. It's really exciting.
Okay, and so I'll finish with mine then. I mean, I think the fact that we're talking so much about the blood biomarkers and how we use them is really exciting, because I think that we've got some that are good enough to use already if we get the context right and the standardization and the collaboration to make sure that there is a consensus. I think that that's really exciting. And I think the next big thing, I hope the next big thing is that a tau therapy works, and maybe in combination with an amyloid therapy proves to be more because we know amyloid drugs are beneficial. And to a patient or their caregiver, that might be really important. But if we can up that, I think that would be kind of really, really exciting. So that's what I'm looking for in the next year. I know there's a talk that I'm just going to go through. I'm really hoping then that I can be right. But anyway, thank you so much. But that's all we've got time for today now. So don't forget to tune in to part 2 for highlights from the rest of the week, which will be filmed very soon. But for now, I'd like to thank our brilliant guests, Dr Ariana Gatt, Dr Ian Harrison and Jess Tobin. I'm Amanda Heslegrave, and you've been listening to the Dementia Researcher podcast live from the ADPD conference in Lisbon. Bye-bye.
The Dementia Researcher podcast was brought to you by University College London with generous funding from the UK National Institute for Health Research, Alzheimer's Research UK, Alzheimer's Society, Alzheimer's Association, and Race Against Dementia. Please subscribe, leave us a review, and register on our website for full access to all our great resources. dementiaresearcher.nihr.ac.uk.